All right. Hello, welcome, folks. I think we're right on schedule to get started. I'd like to welcome everybody here to PEQG 2022, both to folks online and here in person. I'm Jeffrey Rossi Barra. I work on evolutionary genetics of maize at UC Davis. I'm really excited to be here, and I'm honored to be part of the organizing committee for PEQG. This is the fourth PEQG, only the second standalone PEQG. PEQG was started in 2016 by Mike Lynch and other folks, and you'll hear a few words about Mike in a few minutes. We've been really happy with the enthusiasm for the conference. Between in-person and video and online, we have more than 500 attendees, representing something like 27 different countries. And although nobody's really counting, we have more abstracts than the 2018 standalone PEQG. So that suggests that the enthusiasm for the conference is continuing. What I liked most about PEQG and why I was happy to be involved was the science in the conference in each of the times that I've been. It's been really amazing. We have about 70 talks this week, including keynotes from Ryan, who we'll hear from in a few minutes, Magnus and Plenary. We have the oral sessions that you'll hear throughout the week are chaired by these folks, the session chairs. And these are early career scientists that we think are invited to come be the session chairs that we think represent some of the best PE and Q that the field has to offer. In addition to chairing the sessions, they'll each be giving a small keynote on Friday. So there's a special symposium of the session chairs on Fridays, so make sure to check that out. I'd highlight as well the Dalil Nasser Awards. So this is a GSA award for professional development. 3 of these awardees are here at the conference today. They get the award to come present their work. So please, if you have a chance, look these folks up. In addition to the talks, we have a ton of posters, 250, I think, in person and another 100 online. So make sure to check out those posters online. GSA has also baked into the conference a good amount of professional development. I just came from the multilingual networking. There was early career development earlier today, and a bunch of online activities on Friday. And the rest of the week, there's virtual networking. It's on the calendar for during some of the meals, so you can take advantage to network with folks online. And there's on Thursday at lunchtime, there's a Q&A with publishers of Genetics and G3. So if you have questions about publishing, how to answer reviews, what it's like to be an editor, please check that out. If you haven't already, download the conference app. All of this content is in the conference app, including schedule. You can connect to folks. You can post comments on posters. I've already had conversation back and forth with some of the online posters. So it's a good way to connect to the conference at any time of the day and with some of the folks online. And if you're as directionally challenged as me, there's also a map of Asilomar, which can be useful. GSA has also set up a Slack, so you can keep discussing things. You can ask questions about posters. Many of the online posters they'll continue to be online after this week, so you can continue the discussion on Slack as well. Finally, some logistics. All of this is in the website and on the app, but I should highlight a few things. GSA has been committed to make this a safe meeting, and while I know the vaccine requirements and the testing requirements have been a bit of a chore, I'm sure we'd all rather go home with exciting science and not a new infection. So in that spirit, please make sure to wear a mask throughout the conference, even when you're presenting, during the poster session, etc. Obviously, that's not possible during meals, but the meals are available to go. So if you ask for your meal to go, you can grab your meal from the cafeteria and take it outside. There is a code of conduct for the meeting. GSA has a very nicely done code of conduct. Please take a look at it, and we hope that everyone will abide by the code of conduct during the conference. There's information on the app and the website for the code of conduct and how to make an anonymous reporting of violations of the code of conduct as well. Fortunately, the conference is pretty social media friendly, so the assumption is that all of the talks can be tweeted or whatever your favorite social media is. If you want to record a TikTok in the middle of maybe that's possible too. But please don't take photos or videos of the talks and don't share PDFs, et cetera, of people's work because some of this is unpublished material. But tweeting is fine unless a speaker says otherwise. A note to those of you presenting, please check in and upload your slides the day before or online. Make sure you're on the Zoom a few minutes before, 10 minutes before your time to talk, and that for those of you online, the times are in Pacific time. Finally, I'd just like to thank our sponsors, so folks that have made this conference possible, especially defraying some of the extra costs due to COVID precautions. Genetics and G3, Ancestry, the Burroughs Wellcome Fund, and U.S. Biological Life Sciences. And then personally, I'd like to say a huge thanks to the folks at GSA. So when they called me and asked to see if I was interested in helping run this conference, Dmitry Petrov promised me that it would be super easy. GSA would do all the work and I would just get to have fun. I, of course, didn't believe him, but he was exactly right. GSA has done 100% of all of the behind the scenes work, all of the logistics. These folks have been answering emails while on vacation, usually within 15 minutes of me sending an email, as well as keeping us in line and on schedule for everything to happen. A number of these people are here at the conference. So if you see them, give them a high five, buy them a drink, however you want to say thank you for all of their hard work for actually making this happen. So with that, I'll end my brief opening remarks and turn it over to Steve Munger from GSA Board of Directors, who has a few words, and then we can get started with the keynote.
Thanks, Jeff. Give me 1 second. Wow, there's so many of you. This is weird. Great. Good afternoon, everyone. It is really great to see people in person after 2.5 years of craziness. My name is Steve Munger, and I'm Assistant Professor of Systems Genetics at the Jackson Laboratory in beautiful Bar Harbor, Maine. Please come visit us. I've been honored to serve on the board of directors of the Genetics Society of America for the past 2 years. And I can attest to you that our GSA board members, and especially our staff, are committed to supporting and growing this scientific community. On behalf of the GSA, I'd like to welcome you all to beautiful Asilomar. GSA is thrilled to host our annual Population Evolutionary and Quantitative Genetics Conference. But this year, we are especially excited to be reuniting with this community in person. And to all of you attending virtually, we are so happy that you are able to join us for this amazing meeting from afar. A silver lining of the pandemic is that we figured out how to include the hundreds and sometimes even thousands of folks who want to attend a GSA conference, but for one reason or another are unable to get here in person. We now have the technology and the experience to offer the virtual experience alongside the in-person conference. We'd like to thank our dedicated organizers, Chair Jeffrey Ross Ibarra, Jose Crosa, Amelia Huerta Sanchez, and Brandon Ogbanyu for their work in developing this excellent scientific program. We'd also like to thank GSA staff for bringing this conference together, COVID curveballs and all. I cannot say enough about how nimble, creative, and dedicated this staff is. They are kind of shy and would not allow us to put their photos up here, but I have to give a shout out in particular to Susie Brown in the back and Anne-Marie Mahoney, who's not here, but has been instrumental for this conference. They are the primary conference organizers, and we thank them for their tremendous efforts over these past few years that I think we can say have been challenging. This is the first year that GSA has held hybrid meetings, and we appreciate any patience you can muster as we do our best to keep events running as smoothly and possible, both in person and online. Think of this as a new PCR experiment, and we're currently at that stage where we're optimizing our primers and thermocycler settings. So patience is key. Michael Lynch could not be here today, but I want to congratulate him for being awarded the Thomas Hunt Morgan Medal for lifetime contributions to the field of genetics. He is being recognized for his founding of the field of evolutionary genomics, developing the concept of functional diversification by subfunctionalization in duplicate genes, and contributions to evolutionary quantitative genetics and conservation genetics. The award also recognizes his contributions to training and mentoring of undergraduate, graduate, and postdoctoral researchers. In addition to being a past president of the GSA, Michael is a member of the U.S. National Academy of Sciences, the American Academy of Arts and Sciences, the Society for the Study of Evolution, the Society for Molecular Biology and Evolution, and the American Genetics Association. He has served on dozens of NIH and NSF study sections. Within the GSA, he has been a strong spokesperson for population genetics, and he founded the PEQG. In short, he has been an outstanding contributor to the development of genetics as a discipline, and to the GSA and to this conference in particular. You can view Mike's Morgan Medal Award talk online on the GSA Awards seminar page. For those of you who don't know, the GSA is an international professional society for scientific researchers and educators in the field of genetics. Many of you probably just clicked the Join GSA as a Member button in order to save money on registration fees. And that's how I joined the GSA long ago now. And there's nothing wrong with that. We all want to save money, right? But what you may not know is that scientific societies in general, and the GSA in particular, are an indispensable part of the fabric of our scientific lives. They bind us together when the world seems like it is falling apart. Like, for example, when the in-person 4 years in the making 2020 Allied Genetics Conference had to be canceled at the last minute due to the pandemic in March of 2020. That was a very difficult decision. GSA managed to flip the whole thing online without any precedent or example or guide, and they did it in less than 2 months. It was incredible to watch from afar. Just when we were most isolated, GSA brought us together to network and share our science so we could continue to make progress despite being locked out of our labs. Conferences are a key contribution that GSA makes, but GSA also works to advance the field through professional development, community programs, advocacy, and publication in its 2 journals, Genetics and G3, which are published in partnership with Oxford University Press. Many of you know genetics. Genetics is revered for its storied history since its launch over 100 years ago in 1916. Genetics publishes papers that advance our understanding of genetic and genomic mechanisms and functions. G3 launched 12 years ago as a fully open access journal and is the place for high quality, useful, foundational research in genetics and genomics. Both journals are peer reviewed, peer edited, and known for their expert reviews. I have reviewed many of those articles. Clear decisions and overall author experience. I can speak from experience. Plus, manuscripts that are reviewed at Genetics can be transferred directly to G3, and G3 editors often can make a decision based on reviews from Genetics. So it can eliminate the need for re-review and speed things up, which is important, especially to trainees and people who want to get their grants renewed. Last year, the GSA journals launched a new neuroscience series, which some of you have already contributed to, and I encourage you to check it out. Additionally, a plant genetics and genomics series launches in 2023, and we welcome submissions by June 30th, so get those out. Visit the GSA booth to chat in person with Managing Editor Ruth Isaacson and Scientific Editor Sarah Bay about these series, and to learn more about publishing in Genetics and G3. Finally, don't forget to join us at the publishing Q&A on Thursday during lunch in Woodlands, where you can ask all your questions about publishing and peer review. In addition to being a great place to publish, the GSA journals provide training opportunities for early career scientists. In particular, in the peer review training program, over 100 students, postdocs, and early career faculty received formal training manuscript review at Genetics in 2021 alone. Applications for the 2022 cycle will open later this year, and we encourage you to apply. In 2020, the Society introduced the Presidential Membership Initiative, another program aimed at early career scientists. The PMI is one example of GSA's continued commitment to improve equity and inclusion in our fields. The PMI offers free GSA memberships and abstract waivers to awardees. It is designed not only to increase the diversity of our membership, but also to provide a sense of community and tangible positive experiences for the cohort, such as recognition in GSA's Genes to Genomes blog, connecting with GSA board members, and participating in networking events. Last year, GSA welcomed 82 PMI members, and the 2022 cohort will be announced soon. In other efforts to strengthen our scientific community, GSA advocates for shared resources such as Flybase and other model organism databases, whose funding is in jeopardy. I'm sure many of you know that. In 2021, President Hugo Bellin spearheaded a webinar series that drew over 600 attendees and introduced scientists to resources that encourage collaborations between physicians, research scientists, and genome database developers. Currently, GSA's new Public Engagement and Communication Committee is developing educational materials that demonstrate the incredible importance and impact of research using organisms like yeast, worms, flies, fish, and my favorite, mice. These oral interviews and written stories will be made available to GSA members to use in their own public education and advocacy efforts. So stay tuned to the GSA website. I'm sure you all have it bookmarked in social media channels. Of course, so many of GSA's programs and advocacy work support students like many of you and postdocs because you are our next generation of genetics researchers and educators. On that note, I'd like to turn the podium over to Melissa Drown, who can tell you firsthand about opportunities for early career scientists at GSA. Melissa.
Can everybody hear me all right? Great. So thank you so much for the introduction. I'm really honored to speak to you all today about the Early Career Leadership Program, or ECLP, which I joined about a year and a half ago. The ECLP is composed of early career scientists that make up a global network, so we're from all over the world, and we work virtually in teams where we meet to develop ideas and initiatives that are related to the mission of GSA. This platform helps us to gain new leadership skills, build a professional network, and demonstrate our abilities through concrete deliverables. The ECLP is powered by 7 subcommittees, which you can see here. And these consist of co-chairs, members, and advisors who work with various projects on specific goals under the guidance and expertise from subcommittee advisors, GSA Engagement, GSA Board of Representatives, and the GSA Executive Board. I'm just going to briefly outline what each of these subcommittees do so that you can get a sense for what being part of the ECLP looks like. First, the steering committee is made up of co-chairs from each of the other subcommittees with the main purpose to guide the overall mission of the ECLP. The accessibility subcommittee is newly formed and has the goal to understand the access needs of the community and ideate ways to accommodate those through a combination of universal design approaches and increased representation of scientists with disabilities in existing ECLP projects. The Career Development Subcommittee is the committee that I'm a part of, and this supports early career scientists by developing career resources such as workshops and interview series that highlight a variety of trajectories possible within education and research training in genetics. The Community and Membership Engagement Subcommittee works toward addressing concerns of the early career scientist community by building resources and communication pieces. The Communication and Outreach Subcommittee aims to bridge the gap between different scientific fields and between scientists and the public by crafting a variety of informative and engaging articles, and also by managing the weekly newsletter that you may all be familiar with if you're already GSA members. The Multimedia Subcommittee uses multimedia platforms, including podcasts and infographics, to make genetics research more accessible to a broad audience and to promote collaborations in the larger scientific community. And the Policy and Advocacy Subcommittee develops resources related to career training policies and helps connect the scientific community with elected officials and government agencies. Together, the members of these subcommittees compile a variety of resources that are available to GSA members. To name a few, we have the Workshop Wednesday series and the Decoding Life interview blogs, which are posted in the Genes to Genomes blog series that help to demonstrate the diversity of PhD careers available to people with training in genetics. We also have Genetics in Your World podcasts, the weekly newsletter pieces, and other articles in Genes to Genomes that communicate recent research and provide professional development opportunities or to help increase awareness about important issues to our community. Along with that, the ECLP offers a number of training opportunities to its members, which include a professional writing and editing workshop, a leadership and management and action training program, a 6-week leadership, that's a 6-week training program. And then the ECLP and GSA gives us the opportunity to participate, host, or co-organize events in the GSA conferences as well. So with that, I would encourage anyone interested in joining the ECLP to look out for me and other members who have this little yellow thing on their badge that says Early Career Leader. And I'm sure we'd be happy to share with you what the experience has been about and help guide you if you plan to apply to be part of the next cohort, which would be in the fall. With that, I'll hand it back over to Steve.
Wonderful. Thank you, Melissa. I promise we're almost done. So for those of you interested in learning more about the ECLP, Jessica Velez, who is GSA's Early Career Programs Manager and a former ECLP participant herself, will be at the GSA booth Wednesday and Thursday night during the poster sessions. So please stop by and meet her. She's great. I know this will be another great meeting for the population evolutionary and quantitative genetics community. You'll leave here with new collaborations and exciting ideas. Be sure to mark your calendars for the next time this meeting will be held, which is in conjunction with the Allied Genetics Conference, TAGC, being held back in Washington, D.C., where we wanted to be in 2020, March 5th through 10th in 2024, 2 years from now. Now it's my pleasure to welcome back Jeff, who will be introducing our keynote speaker. Thank you very much.
Okay, it's my pleasure to welcome our first keynote to get the science started, Ryan Hernandez. Ryan is a professor at University of California, San Francisco, where he's also the director of a number of institutes and initiatives. Most recently, he is the co-founding director of Propel, which is a really exciting program to provide research experiences for post-bac students from historically underrepresented groups. Ryan has worked in his career on a number of different organisms, rice, primates, microbes. He has a paper on Ebola in snakes, which I don't think he'll talk about today. And I even managed to convince him to get some corn in a recent publication. Most of his work, however, is in humans. What I really love and have long liked about Ryan's work is his ability to sort of move seamlessly from empirical data analysis into theory and through to computer simulation and sort of back and forth between all of those ways of investigating or thinking about population and quantitative genetics. Ryan has investigated all aspects of genetic variation, from thinking about single allele trajectories over time to patterns of variation along a genome to sort of intermediate molecular phenotypes like the architecture of gene expression, to complex phenotypes in humans and the architecture of complex phenotypes and the biomedical relevance of the quantitative genetics of complex phenotypes. In particular, Ryan has done quite a bit of work highlighting disparities in how genomes and genomic data are sampled, how they're applied, and how they're interpreted across diverse human groups. And it's this work on complex phenotypes in humans that Ryan will be talking about today. For those of you who are visiting virtually, or those of you in the audience with the app, you can ask questions during the talk. You can post questions to the Q&A and we can get to those after the talk. Of course, there's also a microphone in the middle, so after the talk, we'll go back and forth between questions from both of those. So with that, Ryan, please take it away.
Thank you so much. That was honestly a wonderful introduction. I almost feel like saying thank you for giving my talk for me. You covered so much of what I do. It is a huge honor to be here giving this seminar and to tell you a little bit about what we've been up to. I will point out, let's see, there's a mouse that should go over there. There it is. You can't really see it. It's a little tricky to see. It's been a long pandemic. This was taken like a year before the pandemic or so. Things have changed. But nonetheless, science, that's why we're here. I am fascinated by human diversity, thinking about phenotypic diversity, how it varies across the world, genetic diversity, also how it varies across the world. And I want to understand the connections between them. As was stated during the intro, I'm really interested in thinking about some of the basic mechanisms of how evolutionary forces shape populations, right? How populations evolve and how those evolutionary forces then transpire to affect complex traits. And one of the concepts that I'll be using is this idea of heritability. I'll use height as an example because it's very easy to imagine. We all have one. And so heritability is really asking the question, how much variation in height among people can be explained by genetic factors, by genetic relationships amongst people? Now, it's important to note that this is a population concept. It's not an individual concept. It's not the same thing as asking how much genetic factors influence height in any one person, right? This is really talking about populations here. And so with height, this has been widely studied. You know, there are many ways of thinking about heritability. One of the ways that people think about heritability is through this idea of broad sense heritability, where we look in very large scale family studies. It might be through twins, comparing monozygotic and dizygotic twins, for example, where we have a pretty good sense what the genetic relationships are amongst people, and we can compare their phenotypes. And through these studies in humans, we estimate that height broad sense heritability is somewhere around 80%, right? Of course, we want to know where in the genome that 80% comes from. And so people have done large scale genome-wide association studies, hundreds of thousands of people. You know, I don't update this anymore because the sample sizes are in the millions now. But the moral of the story hadn't really changed a whole lot. You can look at variants across the genome. You can genotype them. You can impute millions more, and you can estimate the genetic the narrow sense heritability from all of these GWAS loci. And the estimate of heritability comes out to about 60%, right? So there's this difference. There's a gap, which is sensationalized as this case of the missing heritability, right? And I just love this graphic because it shows the crook jumping out the window with a bag of heritability, leaving these sort of modified DNA footsteps in the wake. And so we want to know what in the world is in that bag, right? That's what people have been asking about for a long time. And I think we're finally starting to make some headway to figure that out. And from my perspective, the way that I've been thinking about this is through transitioning into really thinking deeply about deep sequencing, whole genome sequencing in particular. Now, GWAS, I would say, in humans have been hugely successful in many regards. We've identified tons and tons of candidate loci, right? But of course, there are challenges, right? The loci that we identify tend to have low relative risk. They tend to explain only a small proportion of the phenotypic variance individually, right? And they provide the candidate loci. It's hard to get the actual causal loci because they might not be genotyped. And so taking these things together, the implication is that the predictive power of GWAS has often been relatively minimal. Now, of course, we have these things called polygenic risk scores, which are transforming a lot of what we know about the genetic sort of risk for different phenotypes and diseases. But they also struggle, especially once we start thinking across diverse populations, right? And so from my perspective, the way that I want to think about this is through our evolution, right? Human evolution. We know we evolved in Africa and we've sort of experienced multiple waves of migration and have colonized the entire world. And in so doing, we've experienced this crazy amount of population growth in just the last few thousand years. We've experienced more than 3 orders of magnitude of growth, which is quite a lot of growth. And so when we're thinking about this, when we're starting to think about deep sequencing and large samples, we have to really think deeply about what we expect to see, and how does that differ from what we're actually seeing. So I like to always start at that basic level of trying to think about the evolutionary forces and what patterns of genetic variation we might expect to see when we're going to do these large scale studies. And then we actually start observing them. What do we learn new about our evolutionary history? Given that understanding, how do we then sort of translate into developing better powered studies to understand complex traits? And then how do we dive in deep to understand what the genetic factors are that are actually contributing to complex traits? And one of the ways that I tend to think about this is through the action of selection, negative selection in particular. This is just a sort of a sketch overview that we'll all be fairly familiar with. You can imagine each of these gray lines is a chromosome that's segregating in the population, and there are a bunch of mutations that are out there, right? They're segregating. Some of them are at low frequency, some of them are at high frequency, but these are all neutral mutations. But every generation, new mutations enter the population. Some of those new mutations are going to be deleterious, right? What does natural selection do? As far as I can tell, and this is sort of an accurate view of the biological process of natural selection, removing all of those deleterious mutations in a big ball of fire. It might work slightly differently than that in real life, but the moral of the story is that when those deleterious mutations get purged from a population, of course, it's not, natural selection isn't pulling out just those individual deleterious mutations, right? It's entire chromosomes that don't get passed on. And it's that process through which natural selection inhibits the progression of these chromosomes that leads to relatively large effects, both in terms of genetic diversity around the locus where there is actually the arising, the deleterious mutations that are arising and being purged from the population, as well as surrounding regions due to genetic linkage, right, through the effects of selection at linked sites. But every now and then, deleterious mutations can persist for a little while, oftentimes at low frequency. And so if we imagine what patterns of genetic variation might look like under such a process, we could sort of take a step back and think what a neutral model might look like. And there's been a lot of work to develop what the neutral model might look like. This is a histogram showing the frequency of mutations in a small sample size of just say 16 chromosomes. And we ask what fraction of mutations are carried by exactly one of those chromosomes. In this case, on a neutral model, you expect about 30%. What fraction of mutations are carried by 2 chromosomes? And we'd say about 15% or so. And it decreases from there. It's this monotonic decreasing function. This is the site frequency spectrum, right? It's a very important statistic, one that I like to study a lot, because it's really responsive to a lot of evolutionary forces. And in particular, if we were looking at sites in the genome that were actually deleterious, we wouldn't see a site frequency spectrum that looks like this one. We'd see one that looks slightly different. It would look something like this, where there is an excess of rare mutations, right? That's the action of natural selection preventing deleterious alleles from increasing in frequency. But of course, every now and then, you do get deleterious alleles that creep up to high frequency, and it just happens by chance. This is the competition between drift and natural selection, because selection isn't the evolutionary process does not have a deterministic effect. But of course, populations that I'm thinking about don't necessarily just live in this neutral model or this deleterious model, right? There's actually demography going on. There's population expansions and contractions and so on. So if you imagine a demographic process that's also taking place while evolution is acting, while natural selection is acting on deleterious variants, you'd actually see these things compounding each other. So while a neutral model might predict that that in a population expansion, you'd see an excess of rare mutations. If you have natural selection occurring on top of that, you'd see an even larger excess of rare variants, and an even smaller fraction of mutations that are able to reach high frequency. All right, but of course, when we're thinking about populations and evolution and so on, we can't really think of all mutations as having a single selection coefficient, right? There's going to be some variation. Some things are going to be neutral, some things are going to be nearly neutral, some things are going to be slightly farther off on this tail all the way out to lethality, way the heck out there, right? Way over there in Jeff's land. No, I don't mean that you're the source of lethal mutations. I just mean this is the end of the tail. Sorry. But when we think about the and thinking about the patterns of genetic variation that we might see in a large scale sample, right? We might imagine thinking about things and breaking it down by breaking alleles down by their derived allele frequency on the x-axis, going from things that are super rare, say 1 in 10,000, 2 in 10,000, all the way up to things that are super common. And if we were to imagine simulating a population and throwing in mutations at random in, say, a coding region, we would actually expect to see something that looks like this, where about a third of mutations, are expected to be strongly or moderately deleterious. About a third of mutations are expected to be synonymous. There's some of them that are nearly neutral and so on, for those mutations that are super rare. But as you start looking at mutations that are slightly more and more frequent, you see that the fraction of mutations that are strongly deleterious disappears. Even by the time you get to alleles that are 0.1% frequency, those strongly deleterious alleles are no longer present. By the time you get to 10+% frequency, those moderately deleterious alleles are no longer present, right? And so what this means is that when we're thinking about a genome-wide association study where we're genotyping alleles and even imputing them, right, we're only really accessing those alleles that are common in a population that have been identified and are able to be imputed. And we might be missing a lot of these low-frequency, strongly deleterious mutations that are present in a population. And so we want to be able to figure out how do they contribute to a population's phenotypic variation. So we developed a very simple model to think about that. Right? You can imagine there's a SNP out there, single nucleotide polymorphism, and it has some disease propensity. Right? Through some process, this individual that carries this allele might manifest a disease. And it's possible that that disease you're looking at impacts an individual's ability to reproduce and to pass their genes on to the next generation. And so therefore, this SNP might lead to some variation in fitness. But it's also possible that that same exact SNP actually contributes to multiple phenotypes, right? And it's only one of those phenotypes that contributes to fitness. It might not be the trait you're studying at all. Maybe you're studying some very late onset disorder that doesn't have a real clear mechanism for driving fitness. But that allele might contribute to some other phenotypes. My favorite example here is with end-stage kidney disease, which is highly prevalent in African Americans. And there's an amazing story about an allele that contributes to end-stage kidney disease that was actually positively selected in Africa in the gene APOL1 because it leads to trypanosome resistance and was presumably positively selected because of that. But transport these individuals to the Americas where trypanosome infections is pretty rare, and suddenly this allele, this hyperactive allele, ends up contributing to end-stage kidney disease, right? Two very different processes from this one SNP leading to very different things, where the disease you're studying is not contributing to fitness, but the allele frequency of that allele is shaped by fitness effects. So there may be some connection between the fitness effects of an allele and the disease you're studying. And that might be direct and it might be indirect, right? And so what we wanted to do is just think about a very simple model. So we're going to borrow some terms from different papers that have been published. One is to think about a correlation coefficient, right? This is rho from this the Simmons et al. Paper that was published, where you're just basically saying that there's some correlation between the disease effect sizes that the SNP has and the fitness effects that the SNP has. All right. that correlation might be 1 because you're studying some really early onset disease that contributes directly to fitness, but it might also be 0 if it's completely unrelated. We're also going to borrow this term tau, which is going to take a lot of what we've developed in terms of population genetic modeling of the distribution of fitness effects in populations, and try to transform it into something that we know very little about, which is the overall distribution of disease effect sizes, which we just haven't had the ability to really study in much detail so far, right? And so while we have a good sense of what the distribution of fitness effects might look like in a population, although that's still sort of actively being worked on, we have a much better sense of that than the distribution of disease effect sizes. And so we want to figure out if there's a way to transform what we know into what we don't know, right? And then use data to learn about that process. And so given this very simple evolutionary-based phenotype model, what we want to ask is, are rare variants expected to contribute to phenotypic variation? And if so, when, how, and why? And so we're going to consider a couple of different demographic models. One is a simple one called a growth model, where there's a sort of ancient expansion in Africa, there's an out-of-Africa bottleneck, and then there's exponential growth into some non-European populations. We're also going to imagine that distribution of fitness effects, how they enter a population and how they filter through the allele frequency spectrum through the evolutionary process. And we're going to bring those two together to model the distribution of effect sizes based on this evolutionary model, okay? We're also going to vary the demographic model. We're going to imagine this sort of super exponential growth model, or the explosive growth model as we refer to it, to see how those effects might alter the patterns that we see. And one way we're going to think about this is through a plot like this. On the x-axis, I'm again showing the derived allele frequency, and I'm going to call that omega. It's going to go from rare things down here, like 5 times 10 to the minus 4th over here, all the way to common things. And the y-axis is the proportion of heritability explained by alleles with a frequency less than or equal to omega, right? So this is a cumulative distribution function. So I know I'm going to accumulate heritability as I go all the way to the right. So at the end, I'm going to end up at 1. I know I'm going to end up at 1 because by the time I get to 100% allele frequency, I've included all the SNPs. So I have all the heritability that can be explained, right? Question is, how do we get there? If we imagine a neutral model where there's no relationship between the fitness effects of alleles and the disease effect sizes or the complex trait effect sizes, we would actually expect a model that looks like this, right? Where, where we can imagine partitioning, you please stop. Where we can imagine partitioning the allele frequency into ultra rare, I'm just going to use this thing. Apologies for those of you who are online and might not be able to see this. Into ultra rare alleles, intermediate rare alleles, and common alleles. And under this neutral model, the vast majority of the heritability comes from common variants, right? This is the model that I don't know if we can see this pointer very well either. That GWAS is based on, this idea that common diseases are driven by common variants. Common disease, common variant hypothesis is based on an argument basically just like this, where the vast majority of the heritability should come from common variants if the alleles that are contributing to disease are neutral. Question is that a good assumption, right? If we take the opposite assumption, if we say that there's a strong connection or a perfect connection between, a perfect correlation between disease effect sizes and fitness effects, we see something very different. In this case, what we see is that the vast majority of heritability is actually driven by ultra rare alleles. And all of the heritability is driven by these intermediate to low frequency alleles. None of the heritability is driven by high frequency alleles, right? So we end up in this conundrum. You can have a model where almost all the heritability comes from common alleles, or almost all the heritability comes from rare alleles. But for any given trait that we're interested in looking at, we have no idea which side of this distribution they fall on, right? So we have to figure that out. The challenge is that if we vary the demographic model, this picture also changes a bit, right? So this is the African growth model. This is a very simple sort of 2-epoch model. If we imagine that explosive growth model, thinking about, say, like modeling a European population, we see something that's even a bit more extreme. In this case, the this little slope here that we happen to see where there was some accumulation of heritability for these ultra rare and low frequency alleles is really gone. And so in populations that have experienced an explosive growth, we end up in a situation where there's some amount of heritability driven by common variants. And whatever isn't explained by common variants isn't explained at all by low frequency alleles. It's explained by these ultra low alleles, right? So it's either common alleles or things that are super duper 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 rare in a population. And there's very little heritability in between. And that tells us something about how we need to think about studying these traits. Because if we're going to study them, we have to actually study them in a way that's that is informed by what our models sort of predict the data should look like. And so when I talk about genetic architecture, this is really what I'm talking about: the contribution of low allele frequencies and high allele frequencies to the patterning of phenotypic variation in a population. There are many other components of genetic architecture that you can imagine: epistasis, dominance effects, and all those things. They're not included here yet. And so I just wanted to sort of point that out. And the key finding from this analysis is really that, by and large, rare variants are a major contributor to complex traits only when causal variants are deleterious, right? If there isn't natural selection preventing causal alleles from increasing in frequency, then the causal alleles will sometimes increase in frequency, and you'll have a major contribution from common alleles. The only time that that rare alleles really make a substantial contribution to complex traits is when those causal alleles are predominantly deleterious. And so what's also interesting is that this process of demography actually has an effect in and of itself. As populations expand and contract, we know that the site frequency spectrum is responsive, right? The more population grows, the more rare variants are introduced. When populations go through bottlenecks, we know a lot of rare variants are lost, and so proportionally more common variants are maintained. It turns out this has an effect, potentially has an effect on the genetic architecture of complex traits as well. In this simple model that I talked about before, right, we have an ancient expansion in Africa that occurred some time in the past. If we do a simulation and we ask, what is the heritability due to rare variants? This is a small sample of, say, like 500 individuals. And we asked what fraction of the heritability is due to singletons in that sample. What we see is after you go through that expansion, the heritability jumps up. The heritability due to singletons actually jumps up. There's way more singletons in your population, and they're actually doing more. And this is across a range of different correlation values, row statistics. If a population goes through, say, an out of Africa bottleneck, what we find is that heritability also responds. The genetic architecture also responds. So you lose a huge fraction of rare alleles. And so the contribution from rare alleles actually goes down. It's expected to go down. But as the population rebounds from that and goes through this sort of period of super exponential growth, the contribution of rare alleles ramps back up again. And it sort of depends then what type of model you're thinking about in terms of the relationship between fitness effects and disease effects that determines the ultimate fraction of heritability that is due to these rare alleles. But the moral of the story is that regardless of what model we're looking at, there is a response of the genetic architecture to demographic effects. And so we need to think about that. If we're looking at populations that have experienced different demographic histories, we might expect them to have different genetic architectures just because they've experienced different evolutionary histories, which is kind of a logical concept, but it was kind of interesting to find when we noticed this. And so then the question is there any evidence for this? Can we actually see these types of patterns playing out? And so one thing that I'm going to look at now is the idea of gene expression and using gene regulation as a model. In this case, I'm going to look at RNA-seq from these 4 European populations, where we have 360 individuals distributed across the 4 populations. And we also have some whole genome sequencing data from the 1,000 Genomes project. And in the end, sort of after filtering and doing a bunch of analysis, we have about 10,000 unique genes that we can play around with. And so in the land of human genetics, 360 individuals is actually a tiny sample size. But given that this is gene expression data, we have about 10,000 genes to play with. So we have different ways of thinking about how we might model that model that data to try and get a sense for the sort of the spectrum of genetic architectures that might be underlying all of these different genes. And so what we're going to do is we're going to think about heritability of gene expression, in particular cis heritability. I'm going to think about a gene and the expression values for that gene, and I'm going to consider the variants that are within a megabase of that gene on either side as being the sort of primary cis effects, and ask about the cis heritability due to all of those variants on patterns of gene expression. Now, there are several pioneers that have developed ways to infer heritability from unrelated individuals. Some of the common methods are implemented in GCTA and LDAC. These methods require complex optimization procedures that, unfortunately, in my hands, failed oftentimes with these small sample sizes, and just lack of convergence. And so, and we also found some instances where they were biased. We actually went a big step back into the literature and pulled out Haseman-Elston regression, which is a phenomenally simple idea to just look at the correlations in phenotypes amongst individuals and regress that against the correlation in genotypes amongst individuals, and it's based on simple regression, which happened to work very, very quickly, and had no bias or sample size issues that we ran into. And so when we apply Haseman-Elston regression, we think about trying to estimate the heritability of alleles at different frequencies, going from low frequency to high frequency. What we found is this, right? When we look at, again, we have allele frequency on the x-axis going from rare things to common things. And now this is asking about the fraction of heritability for variants that are in each of these different allele frequency things. We find that there is certainly a decent amount of heritability due to high frequency alleles. These are all of the EQTLs that have been identified in humans. They're all out here, right? Those are common variants that are definitely contributing to heritability. But the largest thing, the largest contribution to heritability were actually these guys over here. These are singletons in our data set. Now, you might ask, how in the world do you estimate heritability from, which is a population concept, from singletons, which are by definition carried by only one individual in a sample? And this is where sort of the magic of just trying things and seeing if they work happens to jump in, and then doing simulations to prove that you're actually able to get what you think you should be getting out of it. It turns out you can estimate heritability from singletons just fine. And it boils down statistically. You can sort of write down the equations, and it boils down to just correlating phenotypic correlations amongst individuals with the number of singletons that an individual carries. And the brilliant idea behind that was from some of the collaborators here, which just pointed out that individuals that carry more singletons on average are more likely to have a variant that drives them to be a gene expression outlier. And that's basically the signal that is contributing to this. And so the more singletons you have, the more likely you are to be a gene expression outlier. And that is very informative about the genetic architecture of gene expression. But we have this challenge. You know, our sample size is relatively small, just 360 individuals, and we have this idea that rare variants only contribute substantially when those causal variants are deleterious. And so we had this idea that, well, not all these singletons in our sample size are necessarily deleterious, right? A lot of them, the vast majority of them, probably aren't. But is there a way to try and differentiate those that may be deleterious from those that maybe aren't? And to do that, we thought about partitioning those singletons by the allele frequency that we observed in GNOMAD. GNOMAD is this large whole genome sequencing data set. It's about 15,000 individuals at the time we did this. It's larger now. And the idea is that we can sort of see what frequency are our singletons in this large external data set, right? Some of our singletons are actually going to be common variants in this other data set, and we just happen to see them only once in our data. Some of them are going to be really rare. And so what we did is we then estimated heritability as a function of the GNOMAD allele frequency instead of using our allele frequency spectrum. And this is for only singletons. And when we do that, what we find is that about 80% of the heritability of singletons, the singleton heritability, comes from those alleles that are super rare in Nomad, and in fact, not even present in Nomad, right? The remainder comes from other alleles that are very low frequency in Nomad, and some other sort of things that jump out over here sort of randomly. So this really does sort of corroborate our hypothesis that that rare alleles are contributing to gene expression heritability simply because those causal alleles are actually deleterious and are being maintained at low frequency by negative selection. This idea that rare variants are contributing to complex traits is becoming quite widely appreciated now. There was a recent paper from the UK Biobank, from TopMed, that looked at height and BMI, and what they found in both cases is that these really low allele frequencies are contributing a substantial amount of heritability. Half of the heritability, in fact, was coming from these low minor allele frequency alleles in both height and BMI. And we looked at the UK Biobank data across a bunch of different traits. And what we found was that on average across 72 different traits where we had at least 25 to 50,000 individuals, half the heritability was explained by low frequency alleles, less than 5% allele frequency. Right, so this is contributing to this idea that low frequency alleles could be a major effect that are driving heritability in complex traits. We just have to be able to find it and analyze it. But there's a big challenge in all of this work that I've talked about so far. The challenge is that all of this analysis has been done in a relatively small population of humanity, and those are individuals with European ancestry. It's been widely appreciated now that genomics is failing on diversity, and we've failed to include the broad range of diverse populations across the world. We have this idea that there's this rich history of this evolutionary history of humans, but the way that we typically address this is by sampling this population, that population, the other population. In this case, we can imagine sampling individuals from Africa, from Europe, and from the Americas. Populations that have evolved relatively independently of each other for tens of thousands of years, right? And if we do that, and we genotype these individuals, and we run this through a data reduction algorithm like principal component analysis, it turns out there's plenty of data in the genome to fully separate all these populations, right? You can separate each of those populations into their respective groups. We can identify the European individuals, the African individuals, and the Amerindigenous individuals. But of course, this doesn't necessarily reflect the recent demographic history of the Americas, which we know was inhabited by indigenous populations, colonized by Europeans, who then forcibly migrated a large number of individuals from Africa through the African slave trade. This has resulted in a substantial amount of admixture. And so while we think this sort of basic idea of being able to segregate populations based on what we know from sampling individuals from different continental groups, we think that might hold, and we can identify or label different populations such as white individuals, Hispanic or Latina individuals, Black or African American individuals. That's not the way the admixture works. This is what the admixed genome looks like. This is my genome. And so one thing you'll notice is that there's a lot of colors here, right? But one characteristic pattern is that on each pair of chromosomes, one of them is entirely blue. It's because my mother is white and my father is Mexican American. And so one chromosome is very colored. And so you can see that what admixture does over time is that it results in these mosaics of ancestry, where you have different ancestry compositions at different parts of the genome as you move across. And so there's been a large-scale study. This is to start thinking about admixed populations, which are becoming the majority, at least in the US. It's already the case in California, and it's predicted by 2044 that admixed individuals or minority populations become the majority. There won't be a majority population anymore at that point. And the Hispanic Community Health Study or Study of Latinos, HDHS SOL, is a large-scale data set that has been sampling individuals of different Hispanic Latina populations. And when you take people like me that have admixed genomes and you bring a bunch of us together and you do that same kind of principal component analysis, you don't see these discrete groups. What you see is a continuum, right? All these populations are connected. All these individuals identify as Hispanic or Latine, but they have varying levels of Hispanic, of Amerindigenous ancestry. They have some have large levels of European ancestry or African ancestry. And so the idea that we can try and chop this into discrete groups is kind of a problem. It just doesn't work, right? There's no clear place to cut off these populations to identify discrete groups. And so there's been a lot of work recently to think about this and to sort of highlight this idea that humans are a continuum, and we need to think about that continuum. But even so, even just trying to identify ancestry and trying to identify, say, Amerindigenous ancestry, what is that? What is African ancestry? What is European ancestry? What does it really mean? There's a lot of complexity there. And for individuals who identify as Hispanic Latine, that's actually quite a challenge, because there are multiple different Amerindigenous groups that have contributed to the ancestry patterns that we see across populations. This is a UMAP analysis looking at that same data, coloring it two different ways. One by the ancestry proportions, when we sort of take these three groups, these African ancestry, Amerindigenous ancestry, European ancestry, and you can see that there are gradations here. There are different wings in this plot, where there's levels of increasing Amerindigenous ancestry on the right-hand side. There's increasing levels of Amerindigenous ancestry on the left-hand side. But those actually correspond to very different groups. On the right-hand side, we have Mexican Americans. And on the left-hand side, we have Central and South American individuals. They're actually, there's a decline of increasing Amerindigenous ancestry to different Amerindigenous groups, right? So we can't just necessarily think of Hispanic Latine individuals as one category, right? It just doesn't work. It isn't necessarily helpful. To add some complexity to this, we took a deeper look at the Mexican American individuals who are our largest subsample, and we wanted to ask a simple question, right? How has the patterns of ancestry changed over time in Mexican Americans and individuals who identify as Mexican Americans? And so we decided to plot the Amerindigenous ancestry of each individual as a function of when they were born, from individuals that were born in the 30s and 40s all the way up to the individuals that were born in the 80s and 90s. And we found something that was quite surprising, is that it's not actually constant. Now, admittedly, there's a lot of points here. There's a lot of complexity within this broad group of individuals who identify as Mexican Americans. And so if you sort of zoom in a little bit and do a bunch of lowest smoothing to try and figure out what the patterns are here, we actually see a substantial change in Amerindigenous ancestry over this very short period of time. Now, this is on the order of 50 years, right? We think of human generation time as what, 26 years, somewhere around there, right? This is essentially just 2 generations. And over this 2 generations, we see an average of about a 20% increase in Amerindigenous ancestry over just 2 generations, which is a substantial change for a large population in the like Mexican Americans in the US. This effect was actually, we replicated across multiple cities across the US. We replicated it across different data sets that were sampled independently. We see it across different groups, not just Mexican Americans, but also in other subpopulations of individuals who identify as Hispanic Latino. So this is a broad trend where there is changing or shifting ancestry patterns over time, over a relatively short period of time in the US. But does this mean anything? It turns out we wanted to figure out, does this actually contribute anything meaningful to thinking about complex traits and the health of populations who identify as Hispanic Latino? And so in Mexican American individuals, we correlated Amerindigenous ancestry with a bunch of different biomedical traits. And what we found is that a bunch of them are correlated with Amerindigenous ancestry. We're not the first ones to study this. There have been a few others who've pointed out some of these correlations. But what we did was we were able to show that there are certain traits like height where there's a huge negative correlation. The more Amerindigenous ancestry you have, the shorter on average you tend to be. There are other biomedical traits that are negatively correlated, such as several lung phenotypes, FEV1, FVC. There are also a bunch of traits that are positively correlated with Amerindigenous ancestry. There are some that are really important, things that are really sort of hotly debated, especially at UCSF right now, such as EGFR, which is a measure of kidney function. It turns out kidney function, EGFR, is highly correlated with Amerindigenous ancestry. And so we might need to think about that if we're going to be using these equations in the clinic and identifying what their what an individual estimating what an individual's kidney function might be. And so we still know relatively little about the genetic basis of most complex traits. This evolutionary modeling gives us insights into what patterns of heritability should look like or might look like. And by analyzing heritability patterns across traits, we're able to demonstrate, I think, a major impact of rare and low frequency variants across a large number of traits. And it largely, I think it's largely due to our particular evolutionary history. The other thing is that studying diverse populations introduces fascinating dynamics, which is just super fun to think about. Fun changes in patterns over short periods of time. I think it's just really fascinating subject to be engaged in. But the challenge is that existing tools fail kind of badly. we need several improvements. And while this field of thinking about admixed populations is becoming popular, there are still too few people that are asking these questions. And this is sort of reflected by the demographics, at least within California. This is sort of looking at the proportion of individuals who identify as these different race and ethnicity groups over time, from the 70s all the way to the 2020 census. And what we see is now in California, there is no majority, right? We are a minority majority state, with 35% of individuals in California now identifying as white. But this isn't at all what our graduate programs at UCSF look like, right? Over time, this is just sort of looking at how patterns have changed for our graduate students at UCSF, in particular, those who are from non-URM backgrounds and from URM backgrounds. And what we find is that while there has been some progress over time, the progress hasn't been good enough. And so we wanted to figure out what are the factors that are impacting the ability to recruit and train diverse graduate students. And a number of years ago, faculty at UCSF studied this within one of our graduate programs and asked, how should we be selecting our graduate students? And tried to figure out, is there a way to identify graduate students who've performed really well during their graduate career and those who've struggled mightily? And tried to figure out, are there any variables that from their applications that might correlate with that? This study is confounded by all kinds of things in terms of how you actually define someone as being a successful graduate student versus someone who struggled. Of course, it's biased by the fact that we're only looking at students who were admitted. I wasn't part of the study, but they only looked at students who were admitted. They didn't look at the set of students who were not admitted. So there's all kinds of problems. But the major thing that they found was that the primary variable that correlated with graduate student success was previous research, right? GREs did not really correlate at all. GPA did not really correlate at all. And so the question is, how do we increase diverse graduate students? Well, we have to get graduate students to be competitive for graduate school. How do we get students to be competitive for graduate school? In my opinion, it comes down to training. And how do we increase the training and experience to be competitive in graduate school? UCSF is a graduate student only campus. We don't have any undergraduates, so we can't really contribute to that side of the equation. But what we realized was, especially during the pandemic, is that when labs were shutting down, there was differential access to get research experience, right? And that differential access wasn't going to be felt uniformly across individuals. It was going to be felt more so by individuals with diverse ancestry, those individuals that couldn't necessarily donate time because most undergraduate positions are unpaid. And so we developed this new program called Propel that was introduced, this postbaccalaureate research opportunity to promote equity and learning. And the idea is to train, to provide 1 to 2 year postbaccalaureate research opportunity for trainees from diverse backgrounds, from historically excluded or disadvantaged backgrounds. We provide they're hired by individual labs who pay them equivalent to what our graduate students get paid, so on the order of 40,000+ a year. And we encourage the labs to apply for NIH diversity supplements and engage them in high quality research. And we developed a bunch of activities to go along with this, because just getting research experience is not sufficient. One of the biggest challenges with becoming a successful scientist is actually becoming psychologically ready to have the identity to identify as a scientist, to be able to have the confidence to ask questions, to be able to have the confidence to dive into things that are very hard when you're not sure whether it's going to work out or not. And developing that confidence requires a lot more social and emotional development alongside with the scientific development. And so with this process, we developed this thing called a matchmaking event, where we bring together a large number of trainees that are interested in getting post-baccalaureate research. We take a bunch of faculty who are really interested in training diverse students, but typically faculty don't have the ability to do the outreach to recruit diverse students. And so that's what we do, right? That's what our goal was, to develop that. So far we've done this twice now, and both times we've had over 90 trainees signing up. We've had over 120 UCSF faculty signing up to hire folks. In our first year, we had 11 trainees. This is in 2020. We're now just at the end of our second year. We have 72 trainees. This is rivaling the total number of URM students in all of UCSF graduate programs, which is quite surprising after just 2 years. We've had our first round of students starting to apply to graduate school. There were 13 of them, 11 got interviews and 11 were accepted. 2 of them are still continuing on our program. And importantly, we engage them in all kinds of diverse aspects of science, and trying to think about becoming broad scientists, introducing them to statistics and programming, to thinking about how you become a good scientist by writing, grant writing and things like that. How do you actually formulate a proposal? We even have it set up where they develop mini thesis committees, where they have an affiliate mentor to form that thesis committee, to really give them an experience of being a graduate student before actually attempting to apply to graduate school. And so we're excited to note that we are in the final stages of achieving some foundation funding to help us expand this. And so we're looking at outreach to other campuses that are interested in developing similar types of programs. And I would be happy to talk to anyone who's interested in pushing this further along, so we can expand this across the country. And so with that, I would like to thank you. I am super glad to be here in person. The sort of coming out of our caves has been very exciting, and I look forward to talking to many of you in person. Thanks.
Please feel free to use the microphone in the passage there to ask questions. And I'll take questions online too, for those of you on Zoom. Maybe, Ryan, I can start us off. You presented your model of the sort of proportion of heritability due to rare variants was a fairly simple model of negative selection on a trait. Would that hold similarly for a trait under stabilizing selection? How much would it depend on how recently the population has reached its current optimum? Sort of, what are your thoughts on how that those qualitative results would hold under different models of selection on a complex phenotype?
Yeah, that's a great question. I do think about stabilizing selection a lot. And one of the interesting things about stabilizing selection is that if a population is very near to its optimum, then most variants that perturb it are going to be deleterious. So it's going to drive it to the trait to be, let's say it's too high or too low or whatever the trait might be. And so in that situation, it would be, I think, a very nice intuitive model for what might be driving the evolutionary process here. So I think that's a good way to think about it. One of the interesting things that we've been thinking about recently is, especially when you're thinking about multiple populations, when you have different populations that are in different environments and they have different optima, how does that shift the modeling? And I think in that situation, you have to think about it a bit differently, because in this way, we've only thought about deleterious effects so far. In that case, you're going to have to think about adaptive ones that are actually allowing a population to evolve to some other new optimum, which I think would be super fascinating to work on.
Thank you, Ryan. That was really interesting. I was intrigued by this trend you're seeing in Amerindigenous ancestry, and that was among Mexican American people identify as Mexican American. Two questions about that. First, a technical question that, you know, Amerindigenous groups are undersampled in the reference panels, I guess. And do you think that affects your results? And then maybe more speculative question is, what do you think is going on? Is it different sort of populations moving into where you could be sampled, or what do you think is driving this trend? Purely speculation.
Yeah, sure. So the first question was about undersampling and whether we think there's a bias in our inference of Amerindigenous ancestry. And that could certainly be something contributing. But it does, I think, relate to the second question, which is about the forces that might actually be driving this shift. So one of the initial things that we expected to contribute to this pattern was migration. Individuals, there's a lot of migration from more rural areas to more urban areas, and individuals that are in rural areas tend to have higher levels of Amerindigenous ancestry. It could be that individuals are just migrating from regions that have different Amerindigenous ancestries. That is confounding our inference. It's possible, but I don't think that that's actually driving it. When we looked at what we would expect to see, if migration was driving, we would expect to see these new individuals that are migrating in with higher Amerindigenous ancestries would bring about long haplotypes, long ancestry track lengths. And the models that we looked at just are not consistent with that, right? So there is definitely some contribution of migration from these rural, of individuals with higher Amerindigenous ancestry, but that cannot explain all of our patterns. We see a lot of other things that are going on. One of the features that we did find was that assortative mating is extremely strong in Amerindigenous populations, in Mexican Americans in particular. There's a high degree of ancestry-based assortative mating. Individuals with high Amerindigenous ancestry tend to reproduce with other individuals that have high Amerindigenous ancestry and the reverse. But this by itself, again, doesn't lead to any change in the average levels of Amerindigenous ancestry over time. This is because you have sort of both sides balancing out with their reproductive properties. But when you combine these two, when you have some number of individuals that are migrating in with higher levels of Amerindigenous ancestry that are preferentially mating with other individuals with high Amerindigenous ancestry, and you have other factors that we've observed in sort of more social epidemiological work, is that individuals with high Amerindigenous ancestry tend to have lower socioeconomic status, and individuals with low socioeconomic status tend to have higher fecundity. When you combine that factor with assortative mating and the sort of introduction of the increased migration of individuals from rural areas with higher levels of Amerindigenous ancestry, these three components can actually explain this pattern really, really, really well. And so I think it's a combination of those three factors that are driving it, much more so than our ability to infer the ancestry's track lengths correctly.
Wow, a rich soup of population genetics and sociology and
yeah. Definitely.
Thank you.
Yeah, so first, great talk. I just was sort of trying to in my head, try to combine your two bits of your talk. In sense, the first part, you showed that rare variants can be very important for, like, as a contribution to heritability. And obviously, if you think about population structure, one thing that might mean is that you get very different patterns of rare variants. That very often you have, say, localized variants. And I was just curious if that, like, how would that change sort of your first part? If you sort of now go back and sort of think about sort of more, yeah, say, there are better models of population structure that take more variation into account.
Yeah. Okay. So if I got that correct, you're asking about the role of migration, how the migration
yeah, or just
might
population
contribute.
structure.
Yeah, well, population structure in general. Yes, so there's definitely this idea that that rare variants tend to be population specific, right? And so if you have a population structure in your data set, you're certainly going to have an excess of population structure contributing to your phenotype and actually biasing your inference. This is one of the challenges with thinking about this kind of analysis on individual traits that are super polygenic, because the population structure is going to be a sort of a genome-wide effect, and you don't really have a good way to control for population structure independent of the signal that's contributing to your phenotype. With the gene expression data, what we can do, since we're thinking about cis heritability, what we did was this interesting trans permutation where we just said, okay, we're only focusing on cis. Any trans effects we're going to assume are either population structure or something else. And so what we did was we permuted things. We took the gene expression values from one gene with the genetic data from a separate gene on a different chromosome or far away. And so we did that analysis to try and see, is there any residual effect if we take the genetic variants over here with the gene expression over here? We shouldn't see any, right? And if we do see any, then we're going to assume it's biased. And so we just got rid of all of that, right? So that did a pretty good job of controlling for population structure in a way that we can do with gene expression data, because we have thousands of different genes that we can look at. With complex traits, it's actually much harder because you don't have a good control, a good way to do that kind of analysis. And so I can certainly see it confounding other types of traits, but it's definitely an active area of research to try and figure out how we can control for that population structure.
Okay, thanks.
Hi, Ryan. Thank you. I was expecting when you were showing the gene expression data, because you had European data and African data, and I was sort of expecting that you were going to tell us about the difference between the two because they have different demographic histories. And I was wondering if you expect the effect of SNPs to be lower in Africa because there's less expansion. Does that make sense?
It does. Yeah. And that is something that I've wanted somebody to work on. With a larger data set that we have access to now. In fact, that's part of aim 1 or no, aim 2, sorry, of an active grant that we have that we'll hopefully have some answers to. Right now, we only have simulation results.
Cool. Thank you.
Hi, Ryan. Thank you for the talk. So based on your model selection, you show that the rare the frequency, the variant they tend to explain more heritability based on the negative selection model. In your genetic architecture of gene expression, you really show that the singleton explain majority of the heritability. So it's a difference between singleton and non-singleton. So it seems that there's a drop. So I'm wondering, why is there, what about the doubleton, the rare, like low frequency variants?
Right.
Okay.
Yeah, that's a great question. If I can get these slides to load and go back, the reason is it follows this pattern. And so what we found is that there is a substantial contribution from common variants, but we actually don't expect to see much of any heritability coming from these intermediate and low frequency alleles. It's sort of whatever isn't explained by these common variants are explained by the rarest variants, which are in any data set are going to be the singletons. And so it actually corresponds really well with the predictions from the simulations that we've done. You don't expect doubletons and so on to contribute because by the time you get to doubletons in a sample of 360 individuals, you've already gotten rid of most of the deleterious alleles. So they're just not going to be present at, even as doubletons.
Thank you.
Yeah, sure.
So I have a question about, I guess, this part of your talk as well. So well, I was wondering whether there was a correlation between the fraction of heritability explained by low frequency versus common alleles and the heritability. And if there is a correlation, what that means for the evolutionary trajectories of populations.
Great question. I didn't look at that overall, but I presume that we have this this modeling assumption that essentially is going to is based on a normal 0-1 model. So the assumption is that the mean effect for any of those allele frequency bins is 0. And that there's some variance around that. And so it's a matter of how much variance it is. And so if we look at all variance overall, I would expect that the mean would be 0. For the lowest frequency bins, for the intermediate, for the high frequency, I would expect the mean to be 0 for all variance. The next question is then, of those that are causal, for the causal variance, what is the effect size distribution? And in that case, we expect that that there's a much larger variance in the effect sizes for the lowest frequency alleles, and that the variance goes down, and so you have fewer alleles of large effect as you move up in that allele frequency spectrum. Does that make sense?
So what does that mean for the actual heritability of the trait?
Right. Okay. What does it actually mean? So it depends. So I guess the question is the number of alleles that are causal, because any common allele will have a high population attributable risk. It'll have a high effect on heritability, potentially if it has a large effect. But if the vast majority of alleles that are causal are low frequency, then that's where the signal is going to come from, right? It's just a matter of numbers at the end of the day. And so it depends on which model we're simulating. If we're simulating, a model where there is a tight correlation between fitness effects and disease effect sizes, then that induces this correlation between fitness effects in terms of how deleterious the allele is and the causal effect on the trait. And so therefore you see a very strong correlation between allele frequency and heritability. You see that there's a lot more heritability for low frequency alleles. Low frequency alleles have a wider distribution of effect sizes and so on and so forth, as compared to the common alleles. I don't know if that answers your question.
All right, thanks.
Yeah.
That was really exciting. And one part that was exceptionally exciting for me was the end where you talked about your outreach and the trying to get students involved in science and get them early research experience. And I wonder if you have any hypotheses about the causal mechanism. You can think about all sorts of causal ways where you go from previous research experience to succeeding in school, in grad school and subsequent careers, and I think I can imagine based on those causal models, I would think about different interventions. And I'm wondering if you can think about, or if if you can speculate wildly on what you think is going on and how your program is developed to fill that issue. Does that make sense?
Yeah, absolutely. I've spent a lot of time thinking about this in particular, and I think there's a lot of debate about the weight that graduate programs should apply to previous research, because it can be exclusionary, right? Most undergraduate research is unpaid. I couldn't have done undergraduate research if I didn't have a work study job in the library that allowed me to do research while I was sitting there and in between checking out books. And that's true for a lot of other people. People that are first generation of college don't necessarily even know research is a thing. I didn't know research was a thing until one of my professors told me to go check it out, and I did, and then all of a sudden fell in love and decided to drop the football team. Which was the only reason why I went to college in the first place. And so there are a lot of people like that don't have the exposure to know to look out for research. And so you don't actually seek out a research position until you're in your last year, right? You just start doing research in your last year, you're just not going to have a lot of experience to know that you really, love it and you want to sort of commit your next 5 or so years to it. And so for me, it's the combination of those two things. The fact that most undergraduate research is unpaid, and that a lot of diverse and disadvantaged students aren't exposed to research until it's too late to have a large body of research to be competitive. And so it's both of those things that we're trying to overcome. Thanks. Thank you.
Hi. I was wondering if you partition genes by other measures of, let's say, conservation, evolutionary constraint like PLI, do you see the contribution of singletons being different across different gene categories? And is that consistent with what your theory suggests?
Oh, great question. So one of the fascinating things that is that there is actually there is some overlap in terms of the genes that are under selection themselves and the genes who are under selection at the regulatory level. And there's not actually that huge of a correlation. And so what was surprising is this has been, there's been some work on this in the past that has looked at sort of estimating the difference of selection coefficients that are operating on the coding versus non-coding regions. And so a priori, I actually don't expect there to be a strong correlation necessarily between these two things, because we're talking about gene regulation, we're talking most of the alleles that are contributing to gene regulation are non-coding. And so the evolutionary forces are actually just different from those that are acting on the coding regions. And this is somewhat what we actually see in the data. Now, I can't say that I was able to get a nice sort of picture that was able to sort of tell me like, okay, yeah, these are the set of genes, or these are the pathways that tend to be under selection or tend to have higher contributions of rare variant heritability and so on. Just because the data is incredibly noisy and we see the signals across the genome in so many different places, that we just didn't see a strong correlation in any one of these categories. In all categories, we saw strong examples of rare variant heritability being high and rare variant heritability being low. And so we just didn't really see a huge correlation.
So there are no particular genic features that would stand out for contribution from rare variants, no?
Well, that's slightly different. So if you imagine thinking about what are the factors that are contributing to gene regulation, and what are the factors that are under the strongest signatures of selection, right? Conserved loci, regulatory regions tend to have a large effect. And so we do see that there's a sort of the more we're sort of using this idea of background selection. So looking at McVicker's B, for example, right? You do see a correlation there. So the stronger the sort of genomic loci level negative selection there is, the more of a signal we do see. Yeah, for sure.
Thank you.
Yeah.
Great talk, Ryan. Thank you. So Mike, it's kind of like a follow-up to his question. So it was about, are there any sort of structural features across the genome where you discovered some of these rare variants to be present at higher frequencies? The other follow-up to that is that since these were individual traits, I was wondering if you had looked at sort of patterns of LD across SNPs that were present at higher frequencies across the genome.
Sorry, say that again?
So did you look at patterns of sort of linkage, linked selection effects across loci that might have been present at higher frequency correlated with one particular trait, but also correlated with something else?
What do you mean something else?
So something that's sitting right next to it that might be correlated with other disease traits.
Ah, I see. So thinking about the not necessarily the pleiotropy of one SNP contributing to multiple phenotypes, but where you have different SNPs that might be
Multiple
having different phenotypes,
Correct, different.
effects.
Yep.
And they might be correlated with each other.
Yep.
I haven't looked at that, no.
I mean, I'm assuming you have linkage maps and whatnot, right? Very
Yeah.
detailed ones.
We've
So-
definitely partitioned alleles by those that are in high LD versus low LD. And there's certainly a strong effect of linkage.
Right.
In fact, surprisingly, those alleles that have lower levels of LD, when even within a frequency bin, have a much larger heritability on average than alleles that have higher LD.
Sure. Okay, great.
Thank you. Awesome.
Thank you for your talk, Ryan. I have a question on the last part you mentioned. We're getting a more mixed population, and I'm wondering what kind of demographic model can model those kind of this kind of process.
What kind of
We're getting a more mixed population in a very short time, and what kind of demographic model can model this? Like, what change it? Does it change population effective population size dramatically in short time?
Right. So there are a few different things. So I guess this is so in terms of modeling, are you sort of referring to like simulation modeling, or what, or do you mean some other kind of modeling?
Like if we are getting a mixed population in a short time, I'm wondering, like, before, if comparing the very homologous population we had before, we suddenly got a more mixed population in a very short time. And in the mixed population, for example, I don't know if I'm right, do we have a higher effective population size? And this is not caused by a certain growth of this population, but like a growth of population might take a longer time and also increase the population size, effective population size, but getting a mixed population, I don't know if is it going to increase effective population size in a very short time, and this might also influence the set frequency spectrum that they will expect.
Right,
So-
yeah. Okay, so you're talking about the duration under which admixture has been occurring. Now I get it. So, and the sort of composition of the populations that are contributing to that. Yeah, so when you imagine you have two individuals that have evolved that are in populations that have evolved separately for tens of thousands of years, and they mate for the first time, right? You get one chromosome from your mother, like my blue one, and you get one chromosome that you can imagine the other one was, say, like red, and you have an entire chromosome. Over generations, those things break down into this pattern of this mosaic pattern, where you have very short stints of ancestry that vary across the genome. And so the more generations that take place, the shorter those tracks become. And so you can see that in data for sure, that the more recent the admixture started, the longer those ancestry tracks tend to be. The more ancient that admixture happened or started, the shorter those ancestry tracks tend to be. So you can certainly see that, and you can model that. There's been some really great work to model those ancestry track lengths for sure.
And will natural selection perform differently in a more mixed population?
That's a mystery, right? So, I mean, it depends on what natural selection is operating on. And so if natural selection is a result of a population interacting with its environment, it depends on what the environment looks like. How much of a variance in the environment is there? And how extreme is it? Yeah. So yeah, that's a much more complicated question. But you can imagine that alleles that have adapted for a particular environment in one area are then sort of migrating to a new environment, and you have different gene environment interactions that can certainly spur evolution and natural selection to operate at sort of with a different sort of fitness effect in the new population, simply because the environment is different. So the strength of natural selection would be different.
Okay, thank you.
I think in the interest of time, we should probably end there. Thank you very much for the engagement. You can certainly find Ryan afterwards. Thank you very much, Ryan. A few quick announcements. So dinner's at 6. If you aren't staying here but paid for meal tickets, you can pick them up in the back. If you're talking tomorrow, please upload your slides as soon as you can in the back as well. And we'll see you tomorrow morning at 9 for